Hello everyone and welcome to the Chapter 1 STEM Challenge screencast. In this screencast, in this chapter, we're going to be talking about some very important molecules for life and processes for life. DNA and RNA, very important. They contain all the information to make a living organism. Proteins, the workhorses of every cell. We're going to discover how proteins are made by the cell using the information from DNA and RNA. And then finally, when things go wrong, what happens with mutations? Just a reminder that we'd like you to read and study from your textbook 7.1, 7.2, and 7.3. You will not have to do section 7.4. Okay, let's take a look at these. So the learning objective is to state the central dogma of molecular biology. We'll do that in a couple minutes. We're going to outline the discoveries that led to the knowledge of DNA's structure and function, and we're going to look a little bit at RNA and the three main types of RNA. I'd like you to read and understand the history of the discovery of DNA in your text. That's from pages 158 to 160, and know the major findings and how they found out about DNA from Frederick Griffith, Oswald Avery, Hershey and Chase, Erwin Chargaff, Rosalind Franklin, and of course James Watson and Francis Crick. Watson and Crick are given a lot of the credit for the discovery of DNA, although Rosalind Franklin really discovered one of the key aspects. So I'm not going to talk too much about their discoveries right now, but I'd like you to read that and understand that for the chapter and for the quiz. The central dogma of molecular biology is that the information from DNA, it's pointed in that red arrow here, the DNA is taken, the information is taken by the RNA, messenger RNA, and it's used to make a protein. Proteins, as I've mentioned a couple times, are the workhorses of the cell. Make sure you read and know the central dogma. The double helix that you'll hear DNA described many times is like a ladder. Imagine a flat ladder going up ladder, let's say 10 feet or so. And somehow it's made of rubber and you can twist that ladder around. In this diagram right here, this brown are the sides of the ladder, and the information, the actual coding, is on the inside. This is just a diagram over here. Let's go to the next one. This is a very good short video to allow you to see how the information is contained in the DNA. I'd like you to watch that. One of the things that Erwin Chargaff discovered was that all the information is contained in 4 molecules. They're the DNA bases. Let me go back for a second. I just want to show you the information, the coding for all life is contained in here, and those are called nucleotides. And Chargaff discovered that there are 4: adenine, cytosine, guanine, and thymine, the A, G, C, T in DNA. And he discovered that cytosine always binds to guanine and T always to A. So those are referred to as Chargaff's rules. Here is a picture of DNA. Look at the top of this, the parent DNA molecule, which consists of 2 nucleotides. And notice here's your C, G, G to C, C to C, A to T. This is where the coding is. And in order to get that information off of the DNA, it has to unzip and sometimes duplicate itself, or sometimes have RNA come and get it. Keep in mind that we learned, as we learned in Chapter 5, that cells are constantly splitting into daughter cells from a parent cell. Well, if that has to take place, then DNA always has to replicate itself on a continuous basis, and that's what this is a diagram of. It's DNA copying itself. The Book of You, DNA, this is also a short 4-minute animation that we'd like you to watch. Does a really nice job of giving you an overview of DNA and how the information is contained. There's another molecule that's very important, and that is RNA. DNA is a double helix. RNA is a single strand. Read about RNA. The important thing to know about RNA is there are 3 types of RNA. Look down here. You'll see there's something called messenger RNA, and what messenger RNA is, takes the information from the DNA, which is in the nucleus. The DNA never leaves the nucleus. It always stays in the nucleus with its information. So RNA comes in, takes the information off of that, we'll see that in a couple minutes, takes it out to where the protein is going to be made on a ribosome, and hooks up with transfer RNA. We'll see all of that in a couple minutes. We're going to show you probably one of the best animations that I've seen. We've seen this video before. This is a zebrafish egg, which goes from, in this case, 2 cells. Here is the shell of the egg. Here are the first 2 cells. Actually, probably about a half hour ago, this was 1 cell that we all started as. This is the yolk. So it goes from 1 to 2 cells, and then to 4 cells, and then to 8 cells. Every time those cells divide, it's important that we remember that the DNA has to be duplicated or replicated. The same thing. Duplicated or replicated means the same thing as far as DNA is concerned. That's one of the reasons we put the video in here. Earlier in the course, we talked about the 4 molecules of life. So your cells are made of 4 molecules, and they're really, really important. One of those molecules is carbohydrates. They store the energy. And they allow energy to be created within your cells. Lipids can store it long term if you want, lipids or fats. DNA and RNA, we just looked at them. They contain the information to make us. And finally, proteins. Now, proteins are the workhorses. Most of the processes of cells are done by proteins. So this section on 7.2 is how does a cell make protein? And what we're going to do, it's a complicated process. This is probably one of the more complicated things we'll learn in this course, but I found a really good animation that I think I'll do a voiceover on it, and we'll be able to take a look at that. So the process by which cells make proteins is called protein synthesis. It consists of 2 processes, transcription and translation. Transcription takes place in the nucleus. It uses the DNA as the template to make an RNA molecule. And translation reads that messenger RNA to make a protein. So take a look at this diagram from your text. Up here is the DNA, but it's been unzipped. Notice right here, it's been unzipped to reveal the bases. And look at this triplet code. There's a code here, CAT. The next code, GAC. The code is read by the messenger RNA in those triplets. Here's the triplet is on the DNA. The codon is on the messenger RNA. Notice we see T's up here, but we don't see any T's down here. In messenger RNA and in transfer RNA, instead of having a T, we use a U. So T goes to A, right? A should go to T, but it goes to uracil. So that's the one change that you should know between DNA and RNA. So this is what happens in the steps of transcription. We have our DNA helix, the DNA which is in the nucleus. An enzyme is just a molecule that helps the processes take place. It's a protein as well. Anytime you see the word ASE, that means it's an enzyme. This unzips the DNA, but then messenger RNA is built takes the information off of here. Now, this diagram from your text is okay, but it's not as good as an animation that we found, and we'd like you to see that now. So, okay, what we're going to do now is we're going to make this larger, and we're going to start the animation, and the first time through, we'll let the person who is doing the voiceover, you can listen to her do this.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So there's a lot of information there, and one of the things that you want to do with this chapter is read the chapter a couple times and watch this animation a couple times, because once you watch it 2 or 3 times, it'll start to get much easier to see the way the protein is made. And remember that proteins are made of builder blocks called amino acids. And we're going to show you a chart. And at first it'll seem very difficult to understand, but it's not. So let's look at the top here where it says there are 20 common amino acids in proteins. So think of them as the 26 letters in alphabet that makes every word in the English language. Well, instead of that, we have 20 common amino acids. So let's say the code, this chart right here, all of the different colors are different amino acids. So let's say the code was CCA. Well, if it's C, here's the first base, C, second base, C, and the third is A, then the code CCA codes for an amino acid called proline. So proline, CCA. Let's take a look at another one. Let's say it was ACG. First base is A, second is C, third, we come down here and find ACG. Threonine is the name of the amino acid. So this work has all been done in the 20th century, and these amino acids then get together in a certain sequence to make a protein and are folded. It's very important to realize that how a protein is folded is really important to scientists, because the shape of the protein will dictate a lot of times how it functions in the cell. So let's take a look at this. There's ribonucleic acid. We know that that's a single strand. Here's the codon over here, AUG, and how we read the genetic code. We just did that on that chart. Take a look at this, the characteristics of the genetic code. It's universal, it's unambiguous, and the genetic code is redundant. That means there are a number of codons for a particular amino acid. The animation that you just saw a few minutes ago is in your textbook in this diagram. This is a very helpful diagram. Over here is the nucleus. Here is the chromosome. Here's our DNA coming out of the chromosome, unfolding, unzipping out of it. Here's an enzyme unzipping the DNA. Our messenger RNA is made. Messenger RNA comes out here to the amino acid, out to the ribosome, where the DNA information is brought by the messenger RNA, and then this growing chain of amino acids is the protein itself. So the important keys here, the important players here, are the DNA, the RNA, in this case it's messenger RNA, this is messenger RNA that comes out, transfer RNA here, and all of this takes place when they put together these amino acids. All of these take place on the ribosome. The last section of this particular chapter talks about mutations, and mutations are basically things that go wrong in the DNA when there's copies made, or we expose DNA to environmental factors that can cause mistakes. Now, it's important to realize that not all mutations are bad. Some are bad for us, some are neutral, they don't have any effect on us, and some actually might improve a species over time. But let's take a look at this particular section. So a couple things that we know are bad for us are radiation. So UV radiation from the sun can really damage the DNA that's in our skin. Really, really important. X-rays. Don't get a lot of X-rays. Limit the amount of X-rays that you get, because X-rays can also cause DNA. A lot of people know that smoking can harm your DNA because there are what are called mutagenic chemicals. That means chemicals that create more mutations. But also, a lot of people don't know that things like barbecuing, meats that have nitrates and nitrate preservatives in them can also lead to mutations. Some ingredients in Acme products, especially this one, benzoyl peroxide, can increase your chances of having mutation. And then there are infectious agents like viruses, HPV, and certain forms of bacteria can cause more viruses. Human beings really a lot of times don't know how bad something is, a technology is for them, until way after they invent it and use it. Here's a good example. In the 1950s, if you were a young child strolling along a city street with your mom or dad and you were going to go get shoes, one of the things that the shoe stores would have are these say, hey, come on in here, we have X-ray fitting. And you bring your little daughter or son in and they could look at their toes wiggling in their shoes. Well, that's because they were using this machine. It was called a fluoroscope. Here's a picture of them right there. And they would say, oh, this is going to be, we're going to X-ray fit your shoes. Well, we were doing this for years and didn't realize that the radiation was bad for human beings. So you always have to be careful with new technology and question how we use it. Here's a man by the name of Stanley Watrous and his young family. Stanley Watrous worked at the Limerick nuclear power plant as a bricklayer, but this was before the power plant had any radiation at all. And Stanley Watrous was radioactive. What happened was, in the summertime, he would sleep in the basement because it was so cold, and he became radioactive in his own basement. That's when we discovered that radon gas in some basements, many basements, I should say, in the northeast part of the United States, was leaking into the basements, and that can also cause mutations and cancer. Stanley Watrous. Make sure in your section you read about different ways that chromosomes can be changed. And this is a fascinating video on how DNA, which is damaged, in some cases just through the natural processes, can be fixed by your body. Really well done. Make sure that you read that one. Okay, we ran over a little bit on this screencast, and please read and study, and good luck on the quiz.